Now that experiments have been completed to determine that DNA was the heritable information in eukaryotic and prokaryotic cells, a couple more important experiments were conducted to determine what the heritable information was in viruses. In this video, we're going to be looking at the Hershey-Chase experiment, which used the bacteriophage, a virus that infects bacteria, and the Fraenkel-Contrat-Williams experiment that looked at the tobacco mosaic virus. Let's start with the Hershey-Chase experiment.
Bacteriophages are viruses that infect bacterial cells. In this experiment, phages were labeled with phosphorus to label DNA and sulfur to label proteins. Phage replication involves the phage inserting its genetic information into the cell of the host, and then the outer protein portion of the phage, which is called the phage ghost, falls away. In the sulfur-containing phage, all the radioactivity was contained in the protein, thus no radiation actually entered the cell, and when the phage ghost fell away, all of the radiation or the radioactive sulfur stayed in the supernatant fluid. However, in the phosphorus-containing bacteriophages, the DNA was labeled, and thus the DNA entered the cell as part of the replication, and therefore the radiation entered the cell. After the experiment, the cells were centrifuged down and the results were analyzed. What they found was that in the phosphorus-containing phages, all of the pellet, which contained the DNA inserted into the cells, was irradiated. However, in the sulfur-containing ones, they found that the supernatant fluid was irradiated, but the pellet was not irradiated. This means that in these phages, the DNA was a transforming substance because the DNA was radioactive and it entered into the DNA and it stayed within the cell. However, with the sulfur-containing ones, as soon as the DNA was inserted, the radiation was only located in the phage's protein coat, and when the phage ghost fell away, so did all of the irradiated material, meaning that it was only localized to the supernatant and it had nothing to do with the actual replication of the virus. Although DNA had been proven capable of being the transforming substance in plants and animals, in bacteria, and in some viruses, there was still suspicion that RNA could serve as a transforming principle in viruses as well. Tobacco mosaic virus contains two forms, Form A and Form B. Form A gives distinct spots on tobacco leaves, whereas Form B gives circular shapes. When protein particles from Form B, which I'll draw in orange, were mixed with a strand of RNA from Form A, displayed infection patterns corresponding with infection with Form A tobacco mosaic virus, that is, with the spots. Likewise, when protein fragments from A were mixed with RNA from type B, it was found that the viruses showed the infection pattern of Form B with the distinct circular shaped infections. Thus, it was concluded that in the case of tobacco mosaic virus, RNA could also be the transforming heritable substance. As we've seen more recently with the coronavirus, that is also another type of RNA virus. So we see that DNA and RNA can both carry genetic information. However, protein is not going to carry genetic information.
I hope you found this video really helpful. The concepts and information presented in these videos will be true no matter what genetics class you are taking. However, the concepts presented in this video are referencing material currently covered in Baylor University's coursework. Remember, if you are currently enrolled as a Baylor student, we offer free tutoring services. Our tutoring center is located on the first floor of the Sid Richardson building. You will find all the details you need to know about these services on our website, www.baylor.edu/tutoring. You may schedule a free 30-minute one-on-one tutoring session online through Navigate, or just drop in during our open business hours. For more information about our current services, please visit our website. Thank you.